Bismillahirrahmanirrahim. Assalamu alaikum. Today we will discuss spectroscopy and its instrumentation. Spectroscopy is an analytical technique. It is used to study the interaction of electromagnetic radiation with matter. As the matter interacts with electromagnetic radiation, it either absorbs it or transmits it, and it provides us a great information about its structure and other properties, and many more. Matter can be any atom, molecule, or ion, while the electromagnetic radiation, as we know, it is a transverse wave that consists of oscillating electric and magnetic fields that are perpendicular to each other and to the direction the wave moves. As in the figure you can see, the wave highlighted in green is a magnetic field wave, while the wave highlighted in pink is an electric field wave that are perpendicular to each other and are oscillating continuously and propagate in the direction the wave moves. Let's have a detailed insight into electromagnetic radiation spectrum. Waves have two properties, wavelength and frequency, that determine its energies and properties. Wavelength. The distance between two consecutive crusts or trough is wavelength, while frequency, the number of waves passing through a point per second. Radiation of different wavelengths have different energies. Same is the case with frequency. Wavelength is inversely related to frequency. So different wavelengths corresponds to different frequencies. Long the wavelength, least will be the energy, because wavelength and energy are inversely related. So the electromagnetic spectrum ranges from a very short wavelength gamma rays to a very long wavelength radio waves. Visible region is from 400 to 700 nanometers, where the highest wavelength is of red while the lowest wavelength is of violet. All these waves have different energies, so they also differ in their ways of interaction with matter. Like UV, ultraviolet radiation, it has high energy as compared to that of visible region radiation. So whenever it is targeted on any matter, it causes transitions, while the IR radiation, that's infrared radiation, which has low energy as compared to that of visible region radiation, whenever it is targeted on matter, it induces only vibrations. The electromagnetic waves moves at the speed of light in vacuum, that is 300 million meters per second, which is the fastest speed possible in the universe. The term spectroscopy defines large number of techniques that use electromagnetic radiation to obtain information like properties and structure of matter. For example, mass spectrometry uses electromagnetic radiation to study the mass to charge ratio, while IR spectroscopy, that's infrared spectroscopy, is used to study the functional group of compound. It also uses electromagnetic radiation. Other examples include UV spectroscopy and NMR spectroscopy. These all spectroscopy techniques uses electromagnetic radiation. So what is the basic principle of spectroscopy? The basic principle of spectroscopy is that we shine a beam of electromagnetic radiation of desired wavelength or desired nature, that can be UV or visible, on the sample and study how it interacts, whether the matter absorbs it all or transmit it without absorbing anything. That provides us the information about the properties of matter. Spectrophotometer. It is an analytical instrument that is used to measure the absorbance of sample or matter. When the light falls on sample, it is either absorbed or transmitted based upon the sample's chemical composition. To measure the absorbance, the spectrophotometer is used. Spectrophotometer is actually spectrometer plus photometer. Spectrometer produces light of any wavelength, while photometer measures the intensity of light by counting the photon number. Let's discuss the instrumentation of spectrophotometer. First one is a light source. Light source emits the light of desired wavelength. The light is emitted in all directions, so to direct it into a single parallel beam, a collimator is present. Next to the collimator is a monochromator that works like a prism. It converts the light into its component wavelengths. A specific light of specific wavelength reaches the exit slit from where it reaches the qubits holding the sample. From this point, the detectors measure the transmittance or absorbance of sample. Absorbance is actually how much the sample had absorbed from the incident light, while the transmittance is how much the sample has transmitted unabsorbed. As the transmitted radiation strikes the detector detects the transmitted radiation signals and converts it into a digital display. So next is a description of spectrophotometer instrumentation. A spectrophotometer consists of four main parts. First one is the light source, then monochromator, then qubits, and then light detector. Now we will discuss the detail of four main parts of spectrophotometer. First one is a light source. The function of light source is to provide the light of required nature and wavelength. Different type of light sources are used in different type of spectrophotometers. The light source used in visible spectrophotometer will be different from the light source used in ultraviolet spectrophotometer. So, different type of light sources are used, and the most commonly used is tungsten lamp. Tungsten lamp consists of tungsten filament enclosed in a glass envelope. It is used for visible region because the radiation it produces or emit falls in visible region of electromagnetic spectrum, and its wavelength ranges from 330 to 900 nanometer. Second one is a hydrogen or deuterium lamp. This is used for UV region wavelength, and it ranges from 200 to 450 nanometers. The third one is xenon flash lamps. The light it emits fall in wavelength region from 190 to 1000 nanometers. That conveys that it emits the light or the radiation for both UV and visible regions. Figure shows the xenon flash lamp. Second one is a monochromator. A monochromator accepts the polychromatic input light from lamp and outputs a monochromatic light. It means that it converts the many wavelength light into a single wavelength light. It consists of three main parts: entrance slit, dispersion device, exit slit. Dispersion device, the device that oscillates the light into its component wavelengths. Prism is most commonly used dispersion device that can be made up of glass or quartz. Quartz prism is used for when dealing with UV region, while the glass prism is used when dealing with visible region. In the figure you can see a prism that is used as a dispersion device that converts the common white light into its 7 component wavelengths or 7 different colors. Next one is qubits. Qubits are also known as sample holders, and these are commonly called cell. These are square-shaped tubes that are sealed at one end. They can be made up of glass or quartz. Plastic can never be used to make qubits as it alters the results of absorbance or transmittance of your sample. Qubits should be clear as possible because the slight impurity can alter your results. In the figure you can see a qubit that's made up of glass. Fourth one and the last part is detector. It is the part of spectrophotometer that receives the unabsorbed or transmitted light from the qubits holding the sample and measure it, then induce an electric charge out of it. That is then read out through any digital device or monitor screen. There are different type of detectors are used that includes the photodiode array, photomultiplier, and phototube. In the figure you can see a phototube detector. So that was a brief introduction to spectroscopy and its instrumentation. I hope you understood everything. If you like it, give it a thumb up and subscribe channel.